Southern blotting. What? Southern blotting is a technique used for detecting specific sequences of DNA in a sample of DNA. Endonuclease cuts large DNA samples into smaller fragments. The segments undergo gel electrophoresis, separating the segments by size. The gel is placed into an alkaline solution and a sheet of nitrocellulose membrane is placed on top of it. Weight is placed on top of the nitrocellulose membrane, causing the alkaline solution to be absorbed into the gel, denaturing the DNA and pulling it up into the nitrocellulose and sticking to the surface. A DNA probe is applied to the nitrocellulose, bonding two specific complementary base pairs on the ends of the DNA on the nitrocellulose. A film is placed over the membrane and is developed. This development only shows the DNA samples that bonded to the DNA probes. The final result is a sample where DNA can be analyzed both by the size of pieces in the sample from the electrophoresis, and by the similarity in composition they show from the DNA probe. Where? The technique was invented by Edwin Southern at the University of Edinburgh in 1975, and although its merits were not immediately recognized, Southern blotting was able to identify the genetic mutation responsible for causing the disease, sickle cell anemia, by 1978. It has since been used in laboratories for paternity tests, forensic analysis, and further genetic research. Since Southern blotting enables scientists to compare specific gene sequences, its largest use is in the field of forensic science. Southern blotting made DNA fingerprinting possible, as forensic scientists were able to produce a specific fingerprint sequence, essentially making an identification barcode for individuals. Forensic scientists could also solve paternity disputes, identity cases, and test for identical twins. Southern blotting covers a very large portion of DNA testing techniques. Why? Southern blotting can be used to identify methylated sites in particular genes. It can also be used for a wide variety of other things, such as testing for mutations in certain segments of DNA, paternity tests, building a genetic profile, etc. Also used for determining the number of copies of a gene in a genome and genetic fingerprinting. The mutation test can find the defective genes responsible for causing diseases. On the future of Southern blotting. The Southern blotting technique still has specific uses in creating genetic profiles and being easily accessible, but in many ways it has been replaced by the more advanced biotechnology of DNA microarrays. Southern blotting led to the invention of microarrays, which serve a similar range of purposes, but work with greater accuracy.